Welcome again, everyone. I'm Professor Brett Ferguson of the School of Agriculture and Food Sustainability, and I'm an affiliate of QAAFI. And I've worked together with today's presenter, Professor Anna Koltunow, for the last over 6 years on the High Gain project. So welcome and thank you for joining us. If we could just go to the next slide, please. So thank you. I'd like to acknowledge the traditional owners and their custodianship of the lands on which we meet today. On behalf of the traditional owners, I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and to global society. Okay, so there's just some housekeeping things to discuss before we begin the seminar. The seminar is scheduled to run from 12 until 1. At the conclusion of the seminar, we will hold a Q&A session. If you have questions that you'd like answered, please use the Q&A button at the bottom of the screen, and we'll address them at the conclusion of the seminar. Please remember to use the Q&A button and don't put questions in the chat because we won't be monitoring that. With that, I would like to, it's my pleasure to introduce Professor Anna Koltunow. Her research is focused on the molecular and genetic mechanisms regulating seed and fruit formation and translating that research into useful outcomes from the agriculture sector. Highlights include tools for seedless fruit induction and advancing knowledge in the regulation of asexual seed formation for breeding applications. Recently, she's been leading the High Gain project, which is a multinational project funded in part by the Gates Foundation. The project aims to develop self-reproducing hybrids in both sorghum and cowpea, with team achievements from the project presented here today. Anna is also a fellow of the Australian Academy of Science, and she's also a fellow of the Australian Academy of Technical Sciences and Engineering. And in addition, she's also an advisor for the ARC Center of Excellence in Plants for Space. So with that, I'd really like to let's extend a warm welcome to Professor Anna Koltunow. Thanks, Anna.
Thank you very much, Brett. So hello, everyone, and I too acknowledge the traditional owners and their custodianship of the lands on which we meet here today. So our High Gain project has actually ended, and here in this talk, I'd like to share with you the outcomes of our project. Brett Ferguson is going to have a bit of a cameo role. He's going to highlight a particular legume gene expression tool which his lab has been involved in a bit later on in the project, in the talk. So just to remind you, and I'm sure you all know, that flowering plants typically form seed via sexual reproduction. So they need the gametes undergo meiosis, then there's double fertilization in the female embryo sac, and that leads to 2 compartments, the triploid endosperm and the 2N embryo. And what that does is it leads to diversity in the progeny. But plants can also form seed asexually by apomixis, and that leads to clonal reproduction, so with a fixation of that genotype. But this is particularly rare in agricultural crops, and in the bottom I've got a few examples of some of the types of crops that it does occur in. And the general features of asexual reproduction are no meiosis for gametogenesis, no fertilization for embryo development, and then depending on the mechanism, you may or may not have fertilization for endosperm formation. So what we've been trying to do in High Gain is to harness the asexual seed formation process to induce synthetic apomixis in sorghum and cowpea. And the aim here has been to develop high-yielding, self-reproducing sorghum and cowpea hybrids, so hybrid seed can be economically saved and sown without loss in yield and quality by smallholder farmers in sub-Saharan Africa. Now, in this talk, this just gives an outline of what I'll be covering. So first of all, the challenges in creating sexual reproducing hybrids. I'll just give a brief summary about the importance of sorghum and cowpea in sub-Saharan Africa. And then I'll introduce you to the High Gain team. Without none of our results would have been possible. And then go through some of the data that some of our achievements in developing sorghum self-reproducing and cowpea hybrids. So if you make a hybrid, you use the process of sexual reproduction. You take a male and a female and you cross it, and you've picked those because the progeny of that F1 hybrid outperform the parents. But if you then save and sow that hybrid seed, because of sexual reproduction, you get trait segregation and loss of all that hybrid vigor, and you can't keep it. So our goal has been initially to try and equip the parents with the information that they need. So when you cross them and you make the self-reproducing hybrid, it undergoes asexual reproduction, induces synthetic apomixis, so you have clonal hybrid seed, and you can keep them and save them. So what are the challenges? Well, you've got to switch meiosis to mitosis. So you generate clonal, unreduced, diploid male and female gametes. And we're using a technology that was developed in Raphael Mercier's lab, and it's called MIME. So it's mitosis without meiosis when it comes to the gametes. And we've been doing this by gene editing and targeted knockouts in typically 3 genes, SPO11. So if you knock out SPO11, you have no double-strand breaks. If you knock out REC8, you have no homolog pairing. And if you knock out OSD1, you skip meiosis 2, which is the reductional division. Then we've been inducing embryogenesis in that diploid egg cell without fertilization, and we call that parthenogenesis. And then we try and enable the normal process of endosperm formation for viable seed development. So we try and make fertilization occur in that central cell. Now, in the end, when you build these self-reproducing hybrids, they have to be breeding friendly, economical, and practical for deployment in the developing countries. So cowpea and sorghum were actually domesticated in sub-Saharan Africa, and they remain subsistence crops for millions of smallholder farmers. Sorghum was actually domesticated in Ethiopia, and Nigeria happens to be the largest country in Africa to produce sorghum. It's second to the USA. And in Nigeria, 80% of sorghum production is actually by smallholders. And I do need to mention that there is hybrid breeding now occurring in some countries in sub-Saharan Africa for sorghum. Cowpea was domesticated in Ghana, and Nigeria is actually also the world's largest producer. 32% of agricultural production in Nigeria is cowpea, and there 42% of producers are smallholders. Statistics from 2023 show that the actual yields of those cowpeas are actually 58% below the world average, so that needs improvement. What are the chances of us putting this GM technology into something like cowpea? Well, for those of you who are not aware, there is GM cowpea that is grown in both Nigeria and Ghana, and it has resistance to an insect pest, so it contains the Bt gene, so that has possibilities for uptake. One thing I should mention to everybody is that there is no known hybrid breeding cowpea currently worldwide. So our work with self-reproducing hybrids actually began in 2014 in the first phase of this project, which we called Capturing Heterosis, or CAPHET. And this was led by CSIRO, and we had 6 international partners. And there what we tried to do was actually see if we could just develop any tools to see if we could move the process forward. So a big outcome at the end of CAPHET was we actually showed proof of concept for asexual seed formation in sorghum. The cowpea toolkit was way behind because there wasn't a lot of genomic or any information that you could start, or even promoters or things that you could actually start inducing self-reproducing hybrids. So that toolkit needed building. So phase 2 started in March 2020, and it ended formally in June, actually, of this year. And it's been led by UQ at QAAFI with 6 international partners, and I'll introduce those 2 a bit later. So these were the main 4 aims: improve the cowpea toolkit, and also sorghum and cowpea genomic capability across different varieties, try and build the self-reproducing hybrid prototypes, test them. An important thing there is looking at yield relative to asexual control and see if you can save seeds over multiple generations. That was the big aim. And then hopefully to try and do some confined field trials. So it was a pretty aspirational goal. And also determine the yield potential in hybrids of relevant sub-Saharan African sorghum and cowpea hybrids to see what the magnitudes of possibility were if you were to introduce the trait. So here are our high gain project leaders, and I'm going to introduce the project management team in a minute. So you're probably very well familiar with the UQ team, Ian Godwin, David Jordan. Of course, you've just seen Brett Ferguson, and of course, Emma Mace. Of course, David and Emma are out at Warwick, based at Hermitage. And then our international partners are Corteva, which is a multinational seed company. Mark Albertson at the start of the project was the leader on the Corteva side, and Marissa Simon has been working with us for the last 3 years on the Corteva side to help the project along. We have Jean-Philippe Vial-Calzada from Sinvestov, who's based at Lanquebio in Mexico. And then Peggy Osias-Akins, University of Georgia. Uli Gross-Niklaus, University of Zurich. Andreas Huben at IPK Gardesleben in Germany. And Takayoshi Ishii in the University of Tottori in Japan. So the management team comprises me. Oh, sorry, I should actually say that the skill sets of all these people are really diverse, from cytology to breeding and genetics, and I've listed them on the slide for you to look at. And this was really essential for the team to actually share ideas and help with the workload. And they proved very collegiate, and that was really a big part of our success. So the management team includes me and Angie Strelo, our high gain project manager, and Eric Pham, who has been helping us a lot with the financials. And I couldn't have done anything without these two guys. So moving forward. Just to give you so we also had, so Vishukla was our Gates Foundation coordinator. We had a fantastic scientific advisory board. Luca Komai, Kirsten Bett, and Greg Grabetsky were the last members of the team. We had Tony Kutchin from the Danforth Centre, Jim Weller from the University of Tasmania. And we also had some project advisors to sort of help us on with sort of general breeding aspects, Mark Albertson and Arp Schnittke from the University of Hamburg, because he's an expert in meiosis and various cell division capabilities. High Gain also has a commercial committee. It still has one, actually. And this actually comprises people from all the different agencies that are part of the team. And what they do is they look after whether or not we have to patent anything, and the way we might go about that. Under our commercial agreement that we have on the project, the Gates Foundation expects us to protect what we can that comes out of the project so that the outcomes of the project can be delivered to smallholder farmers without being blocked. So the way that we've been doing that is Corteva has first rights for patenting, but if they don't choose to do so, then other agencies can do it, or we just make it publicly available. And it's been a valuable resource for decisions that have been made in terms of commercial activities. So here is a panel of some, but not all of the members of our High Gain Lab and field team. These are all the people that enabled our outcomes, and I'm sorry if your photo is not on there. But so over the period of time, we had about 106 members, and the numbers of the people as postdocs techs are listed here. So thanks to everyone here for enabling our outcomes. So what have we done in sorghum? Well, we have established proof of concept for self-reproducing sorghum hybrids. This was done in 2 hybrid backgrounds, 623 by 430 and 623 by Messia. Messia is an African variety. And this was published in the Plant Biotechnology Journal, so it's accessible. You can look at the outcomes, but I'd just like to summarise some of them for you. So the building blocks we used, just to recap, we use mitosis instead of meiosis to eliminate mitotic recombination, and that should be meiotic recombination. Sorry, and meiotic reduction to produce diploid clonal male and female gametes. So we knocked out SPO11, REC8, and OSD1. SPO11, REC8 eliminate recombination. OSD1, if you knock it out, it avoids meiotic reduction. Yeah. So what does that sort of look like if you start to do some cytology? This is looking at male myocytes. A to D represent the normal events of the meiotic process occurring. And then here in this set of panels from E to H, if you knock out SPO11 and you compare the end result, it looks a bit of a mess because there's failure in recombination initiation and there are defects in synapsis and segregation. SPO11 is lethal on its own in the male and the female. And then here you can see the REC8 mutation. So you see errors in chromosome pairing and segregation. And this just also leads to lethality. Now, we came across a bit of a problem with OSD in sorghum. It actually has 3 copies, OSD 1, 2, and 3. Work showed that OSD2 knockout has no apparent phenotype, but knockouts in both OSD 1 and 3 are required to prevent meiotic reduction. So basically, you had to knock out the SPO11, REC8, and the appropriate mutations had to be obtained in OSD1 and OSD3. For parthenogenesis induction, we utilise the capability of Peggy's lab in Georgia. They actually discovered the parthenogenesis could be induced by a gene from an apomictic grass, Pennisetum. And this particular gene is an ASGR baby boom-like gene. I'm just going to keep calling it PAR. And we used parthenogenesis induction by targeting that gene to the egg cell via a maize egg cell specific promoter, which worked. So Corteva used 2 different methods. Basically, these 2 methods just rely on transforming hybrid embryos. And they generated the hybrid embryos in 2 different ways. This is Marissa's 2-step method. First of all, the parthenogenesis gene was introduced into the male, then crossed to the female 623, isolate the embryos, do your transformation, and you transform with a construct that actually enables the gene editing, and then select your progeny. The 1-step method, basically you do your cross, and here this work was done using Messier as the male parent and TX23 as the female. You cross, obtain your embryos, and you transform with a construct containing everything that you need to induce self-reproduction, and then you try and find your progeny. It worked. This shows you, this early on, Marissa selected 4 T0 events, and then she looked at the T1 progeny, SNP markers across the 10 chromosomes, which is what this represents. And basically, the parents, if you've got any of the parental DNA from the dad, it'll be in blue, and from mum, it'll be in red. And as you can see here, it was all yellow. And the yellow, of course, is showing you the hybrid phenotype, so it was successful. So in the cross 623 by 430, she was managing to carry that through with this genotype preservations for 6 generations, and there were 3 glasshouse generations for the 623 Messier cross. So it's all very, very well and good, but how does this compare to your sexual hybrids? Because that's pretty important. The vegetative growth of the self-reproducing hybrids was similar to the hybrid controls. The asterisks here are the actual hybrid controls, sexual hybrid controls. But there was a bit of a problem with seed set. It's reduced in the self-reproducing hybrid. So this is E is the sexual reproducing hybrid. Sorry, D is the sexual reproducing hybrid. Oh, sorry, just getting confused. D is the sexual reproducing hybrids. I'll start again, Anna. D is the sexual plant and E is the sexual reproducing hybrid. I'm getting tied up here. Now, what Marissa and team found was that a single in-frame change giving one functional allele in either OSDL1 or OSDL3 improves seed set in the self-reproducing hybrids. If you knocked them both out completely, it was terrible. Didn't work. So OSD1 and 3 expression and activity is important, and this needs further investigation to get the right set of mutations. She also observed that there was a reduction in seed germination, and this is shown here in G. And you can see the levels are reduced. So you want good germination, and you want it to be as good as a sexual control. So one of the things that UQ did, there were no field work done by Corteva. So Ian Godwin led all of our confined field trials out at Gatton. There were 2 lots of them. The first one investigated the stability of the parthenogenic component. So the question was asked, well, is it stable? It looks okay under greenhouse conditions. How does it work out in the field? Now, the greenhouse rates by Corteva in a TX430 background containing a hemizygous copy of the PAR gene gave 35 to 40% in the plants in terms of expression. It's probably got something to do with where the gene lands because it's a random integration. And the maximum parthenogenetic capacity that you expect in a hemizygote genetically is 50%, so it hasn't really attained that. So we obtain lines of these plants from Corteva. Just remember, meiosis is intact in these, and Ian and his team planted them out at Gatton. And they actually observed the same parthenogenesis. They actually observed the same parthenogenesis expression as was observed in the greenhouse at Corteva. Of course, it was also these plants were also maintained in the greenhouse at UQ. The other thing that was done was this particular line was crossed by Tracy Shady, actually, out at Warwick, into other backgrounds of sorghum. And their parthenogenesis expression was also observed, and it was fairly stable also in one growing season. So this component seems to be stable under confined field conditions and in different backgrounds. So what about the performance of self-reproducing hybrids out in the field? This is also work done by Ian and team. Now, here the strategy of actually generating the hybrids was under a crossing strategy like we had proposed initially, where you equip the parents with the components and you do the cross. So what happened was this was done in Messia. So the Cas9 gene, which actually does the editing, had to be introduced into Messia. You had to introduce the parthenogenesis gene. That's what this nomenclature means. So this parent is essentially sexual. And then here in the female, you've got the guide RNase for Mimi. It was quite a complex cross. I've just done a simple representation here so that you can see what happens. And then when you cross these two, you've then got to, you get a self-reproducing hybrid, but you've got to find it. So you do PCR and sequencing to make sure it's got baby boom, make sure it's got good knockouts in REC8 and SPO11, and this will generate various knockouts of in OSD1 and 3. It proved very labour intensive to identify the plants. It's not going to be easy for people to do this unless they're very good at molecular biology. The team grew the plants in the greenhouse for a few generations, and then they planted them in confined out at Gatton under net. The growth of the plants appeared to be similar to sexual, certainly in the early stages. Here's the sexual, this is the self-reproducing hybrid in the centre. And basically, these experiments in the field started towards the end of last year, and they've just finished. And the data is still being collected. This gives you a general representation. Basically, in both glasshouse and field set, and in the field, seed set is lower than the control. So these are a couple of plants that have that gave good results that also show relevant mutations in OSD1 and in OSD1 and OSD3. But the seed set is still lower, so this still needs improvement. In the field, the plants were assessed for many things. There were drone flights for biomass, and here we've been working with the Australian Plant Phenomics Network, Chris James and team, to sort of do those kinds of flights and things. And one of the things they're looking at as part of their work is to try and perhaps develop imaging for examining seed yields from panicles in sorghum. And of course, the team is doing the laborious work of looking at and counting what the seed yields really are. And that will help ground truth some of the algorithms that they're writing and for some of the imaging. So it's been pretty good to work with them as well. Thank you. So what's the yield potential in hybrid sorghum in the 4 races of, so if we actually look at crosses made over the 4 races of sorghum that are grown in Africa? Commercial sorghum, I believe, predominantly relies on cordatum and QAAFI race crosses. And I believe the QAAFI female, it's easier to introduce cytoplasmic male sterility. But these 4 races, cordatum, QAAFI, Dura and Guinea, are all grown in sub-Saharan Africa. So what is the magnitude of heterosis? So David Jordan and the Hermitage team laboriously did thousands of crosses using a chemical male sterility agent, TFMSA, and they did it in various ways. They did crosses between local adapted varieties, and they also did crosses between some improved by local varieties. So in about 750 crosses in multi-environment trials, they assessed grain yield, grain number, biomass accumulation, and growth rate. And there was clear heterosis, with grain number being the primary driver of yield gain. And they found that elite by locally adapted hybrids produced about 34% higher yield than just doing local and local. So the recommendation from David and his team for strategies going forward is to integrate elite germplasm into locally adapted backgrounds as a short-term outcome, and then for medium to long-term outcomes, develop new heterotic tools based on local germplasm, exploring interracial combinations. So just a short summary of what's been found in sorghum. Heterosis is high in sorghum hybrids and crosses involving parents across the 4 races. We have generated self-reproducing hybrids, which are stable over multiple generations, certainly in the greenhouse. But we need to improve the seed set yields to approach that of the sexual hybrid controls. And we need to make sure that seed germination is equivalent to the sexual hybrid control. So what about cowpea? We did a lot of work to improve the cowpea toolkit. First of all, in most of capturing heterosis, transformation frequencies for cowpea were less than 1% the efficiency. This was improved by work in Corteva, which has been published, anywhere between 8 and 50%, 56% in different varieties, and it also works in hybrids. It's a terrific method. The Totori team developed a system for inducing male sterility for cowpea hybrid production that's been published, and Totori also developed a simple speed breeding protocol, so you can grow 8 generations a year in the glasshouse instead of 3. An extended pangenome was developed for cowpea. This was work done out at Warwick with Emma and Sophie. And now cowpea has a closed pan genome. They added 20 genotypes to the existing set. So you don't need to add any more to improve the knowledge of the diversity there. They also did an amazing job of analysing cowpea global diversity in 10,617 accessions. Pretty cool, eh? The distribution published a lovely paper just recently, and so basically they've also deposited 1,085 accessions, which represent the global diversity of cowpea in the Australian Grains Gene Bank, so that people can access it publicly. Cell-specific promoters have been developed, laser capture cells and whole tissue transcriptomes. This is combinatorial work by the people that worked in the cowpea objective, UQ, Lankebio, UGA and Zurich. And then, of course, because there's no current hybrid breeding programs, we had to evaluate the heterosis potential in cowpea. So is heterosis good enough that warrants building self-reproducing hybrids in cowpea? And this was work also by Emma and David and the team out at Hermitage in collaboration with the Lankebio people and UGA and Totori. So Totori did work on a Japanese set of germplasms in their growing conditions. What UQ, UGA and LAN did is they 45 parents representing a good mix of the global diversity were crossed. And the F1s, so these were done in Queensland, USA and Mexico, and they were grown in these locations to sort of look at the performance. The hybrids yielded more, they flowered earlier, they had increased biomass. The average best parent heterosis ranged from 12 to 20%. Seed yield was the major gain. Seed number drives increased yield. So it's not the number of seeds per pod, it's the number of pods per plant. And over 75% of F1s outperformed the best parent. And you had relatively good phenotype conservation at these three different geographic regions. So obviously the level of heterosis in cowpea is terrific, and it warrants the further development of self-reproducing hybrids. Now, what I'd like to do is cross over to Brett Ferguson now, so that he can tell us about VigExpress.
Okay, thanks so much for that, Anna. Yeah, so I'll just give a bit of an update or explanation of the VigExpress app, which is an app that we developed. So basically, VigExpress stands for Vigna Expression. So it allows us, it's a tool that we developed to visualize basically gene expression in cowpea. And so we found this really useful to help identify new genes of interest and also to identify homologs of genes from other species quickly in cowpea. And we were able to use this to generate a number of new molecular factors and tools for cowpea and for legumes in general. So this is just a screenshot of our recent preprint that came out in bioRxiv. It just came out last Friday. And I'd just like to point out, in particular, Dr. Honan Su, the first author of the article. He's a research fellow in QAAFI who drove much of the work that I'll mention here now. So we could just go to the next slide, please. Okay, so basically when HiGain started, one of the things we did was to gather a number of RNA-seq data sets or transcriptomes that were available. So there were a number of really nice data sets from whole tissues that we were able to access. This includes key parts of the plant like the leaf and the root, but also from the previous phase of HiGain called capturing heterosis, there were a number of samples from whole reproductive tissues that we had access to. And this is just illustrations of a number of the tissues that we had that we could use as data sets. And if we could just go to the next slide then. And in addition, we were very fortunate because we had access to RNA-seq data sets from specific reproductive cells and tissue types. These were collected via laser capture microdissection and are rare. Like I don't know of these being existing in other legumes or many other plant species in general. And again, these are just examples of illustrations of the different samples that we had. And so having these cell and tissue types that we've collected, basically, if you've dealt with RNA-seq data sets before, it's fantastic to have the raw data, but it can be a bit clunky to use. And so what we did was to develop this visualization tool so that it made it a little bit more user-friendly to be able to see and analyze the data that we had. And so that's where VigExpress comes in. So it enables the visualization of gene expression across a range of cowpea cell and tissue types. And it can be based on the gene ID or the gene name. You just type it into the app and it will show you then, or allow you to visualize the expression. So this is really useful to identify new genes, but also homologous genes. So if we had a gene from Arabidopsis or another legume or any plant species, you could then quickly search for it and find its expression pattern in cowpea. So in the app, just to walk through some of these pictures, you see the bar graph. At the bottom of the bar graph is the different cell and tissue types. And the bars in the graph are showing just how much expression there is in each of those samples. The more red it is, the more expression there is. And if it's yellow, it's quite lowly expressed. So in this particular example, this gene is expressed fairly highly throughout the whole plant, and in particular in the seed. And that's what the graphic at the bottom is showing, is the specific tissues that it's expressed in. In addition, you can enter in a number of different genes of interest, and this can be useful to look at gene families. And so that's what we're showing. The other illustration there on the right, this is a heat map, and you can look at various genes that you've entered. It can be a gene family, it can be whatever candidates you're interested in. And this allows you to then visualize their expression patterns. So this can be really useful if you're specifically looking to target a gene to a specific tissue or cell type, but also if you just want to look at families and quickly identify what is likely a homologue of your gene of interest. So it's pretty simple and user-friendly, and we've made it with open source. It's open source so that you can add to it, and it's very adaptable. So as new RNA-seq data sets become available, they can be incorporated into the app. So next slide, please. Yeah, and this is just some examples of new tools that we developed. So one of the things we did was to identify really robust reference genes that you can then use as housekeeping genes for real-time PCR. We also identified key components for genetic transformation and genome editing, such as promoters to drive antibiotic resistance, additional promoters, which could either help in the selecting, we fuse them to reporters. You can use that to select transgenics that were successful, or you can use those, if they're cell-specific, say, you can use them to drive a gene or compound of interest to that cell type. And of course, we found terminators and other things, and we were able to use those to develop new vector backbone for genome editing, which we then used in the paper to, we validated that, tested it, and showed the utility of these backbones by doing CRISPR in the paper itself. Yeah, so basically legumes exhibit a lot, especially the crop legumes. They exhibit considerable amounts of genetic synteny. So often what we find, it's not always the case, but it's very common. If you find a gene in one legume, such as soybean, you can often find a homologue in cowpea, and its expression pattern and often even its function will be quite similar. So this is really useful then, because with VigExpress, we can then take genes that we know from other legumes, especially the crop legumes, and we can quickly identify them in cowpea. So in this particular example, you can see in both the aerial parts of the plant and in the reproductive cell and tissue types, it's very yellow. It suggests there's almost no expression in those tissues. But if you look in the roots, it's quite red. And then we look at the bar graph below, that's confirming that the root tissue is showing very, very high expression. And we were able to validate that with real-time PCR, where basically this gene's only expressed in the root and we didn't find it really anywhere else. And that makes sense because this gene that we looked for was a known gene from other legumes that's key for legume nodulation. So it's called nod factor receptor. Its role is to function in the root, where it, as a receptor, it perceives the rhizobia produce nod factor compound. It's a lipoketooligosaccharide, and it allows the legume host to identify compatible rhizobia bacteria When it does that, it triggers nodulation pathway. So this has been really useful to be able to identify these homologous genes in cowpea and vice versa. You can identify genes in cowpea that you could then probably go back to other legume crops and find that their expression and function can be quite similar. So it's become quite useful for our group in that sense. So with that, I'll pass it back over to you then, Anna. Thank you.
Thank you very much, Brett. So anyone that's interested in that tool, please connect with Brett and Hernan. So, okay, so let's switch to cowpea. Can asexual seed formation be induced in cowpea? Remember, at the beginning of the project, we hadn't even been able to do that and test it. So if we look at the individual components, MIME initially, so avoidance of meiosis, it can be induced in cowpea. So basically, there was knockouts of SPO11, RECA, and OSD1 used, and this shows what you expect to have if you have MIME is if you look at the T0 plants that produce T1 progeny, when you grow them up, you expect them to be tetraploid after they self and seed. And that's because you have 2N male gametes and 2N female gametes fusing to give you a tetraploid. So here shows what you expect with flow cytometry of a normal diploid if you look at their leaves, and here is the peak of a tetraploid in two different T1 lines. So MIME was working successfully, but its efficiency needs improvement because the number of seed that you got wasn't as what was expected, so this needs more work. So this work was actually completed by the Lankebio team actually to create a full MIME knockout. The next thing was we had to try and induce parthenogenesis. And I have to say that this was really hard in cowpea. It's not the same as occurs in the monocots. We actually spent a lot of time using different approaches, which I won't go into, over a couple of years with members of the team to try and see if we could induce parthenogenesis. The problem is you could get the embryo to start growing, but then you couldn't get viable endosperm formation as what you actually get when you in the monocot in sorghum. So one of the approaches that the Lankebio team tried was they actually targeted the cowpea parthenogenesis gene to both the egg and the central cell. Then it was possible to get central cell fertilization with the incoming pollen grain, and that enabled viable parthenogenetic embryo formation to maturity and then endosperm development. So you could germinate these haploid seed and you get your haploid plants, which of course aren't terribly happy, and they don't set seed because it's not possible. But it actually showed proof of concept of parthenogenesis, and this has actually been published. There's a bioarchive article on it, and actually a provisional patent's been filed on the way that this was done. So there's the DOI for that. So what happens if you combine the MIME components and with the parthenogenesis components that have just been described to you? Well, this came towards the end of the project with sequencing. What the Lankebio team did was introduce this into a hybrid background, actually, and successful induction of asexual seed formation was found by sequencing. And of course, unfortunately, the efficiency needs improvement, but at least the proof of concept has been generated, and this is a first for legumes. So if I can just summarize some of the things I've talked about, and what this tells us is self-reproducing hybrid induction is crop specific. It's quite different in the way that you have to do things for sorghum and cowpea in terms of getting expression of the components. We have high levels of seed yield in cowpea and sorghum hybrids, so it further supports the effort to build self-reproducing hybrids for smallholders if the project continues. Sorghum self-reproducing hybrids have been generated. Sieves have been saved over multiple generations. We have demonstrated asexual seed formation in cowpea. Still need to demonstrate genotype preservations in various hybrid combinations, and check it over multiple generations. Efficiency is a big thing. We need to, our target is to actually have self-reproducing hybrid seed set efficiency, which is at least 95% of the sexual control. And we don't really have that, for example, for sorghum at the moment. It seems to be about 56 to 61% of the control. And then we also need to have germination highly efficient. And then, of course, if you have something like this, which is the 95% target is what you'd call a pre-commercial phenotype. If you can actually generate that, then you can work on the installation of the trait effectively so it's breeding friendly. The other thing that's got to be is economic. It must be economical for deregulation, and it's got to be practical for deployment. So there's still a fair bit of work to be done. We aren't the only ones that have been working on self-reproducing hybrids. There's been tremendous success in rice. So a while back, the Sorghum lab in Davis showed proof of concept for asexual reproduction in rice. And of course now hybrid rice has been developed with the capability for self-reproduction. And they've got it at pretty high efficiency. They had to modulate some of the components, different from the SPO11 Rec 8 things that we've been using. And they've actually now been conducting field trials, and they're getting high seed set yields. So it's possible, and it's working well for hybrid rice that they've got in China under test. So I'd like to thank you all very much for listening, and of course thanks to our entire national and international team for enabling our project outcomes. And so we can open up now for questions, and I think Brett's going to manage that.
Yep. Thank you very much, Anna. That was wonderful. Yes, so if there are any questions, I encourage you to enter them into the Q&A. There's a button down at the bottom of the screen that you can enter it into, and we'll be monitoring that. So you gave Anna a bit of I guess, future directions where you think it's going. Could you give a bit more, maybe expand on that and where you think the field, not just high gain, but where do you think this is headed in future?
So at the moment, there appears to be a focus on self-reproduction of hybrids, but it's still got to expand a little bit more into the eudicots. I think there's been some work that's being done in tomato, so that work is continuing. I think, though, if it was a useful technology, what it can be used for is just fixing genotypes when you're doing plant breeding. Instead of having to, so of course, as you know, I mean, I'm not a plant breeder, so I apologise to those of you who are out there. But as you know, you do a cross, you select the variety, but you've then got to do back crossing to get it stable so that you can use it and sell it. And that might take several generations of back crossing, 6 or 7 or whatever it is, which takes a long time to get things to market. If you can use this technology to fix the genotype you want, you could get things, you could actually fix that genotype in 1 or 2 generations, which would, increase genetic gain, I think. So it's not only for hybrids, it could be potentially used in breeding.
Yeah, exactly. And of course, High Gain had a focus with the Gates Foundation on sub-Saharan Africa with major crops in Nigeria and sub-Saharan Africa, but it's not limited to that. And even like in Australia or around the world, this technology can basically, I guess, tap into things where there's a lot of heterotic potential, but it was maybe a challenge to do the crossing or other things where it can be done, but it has to be redone and redone and redone. And so I could see this also being really useful for tailoring strong hybrids for different regions instead of just having the hybrid.
Well, if it can work effectively, yes, you can, especially if you sort of think about climate change and that we've got to adapt our plants quickly to the effects of climate change and get crops out to specific regions. So it could actually speed that process up a little bit. Um, and breeding companies aren't adverse to this. I mean, we have had Corteva involved, Pioneer Corteva involved from the start. And of course, their applications are probably are directed more towards maize and soy, for example. One of the things with the collaboration with the funding from the foundation is that the aim of the foundation is to target opportunities for smallholder farmers so they can benefit. There isn't a restriction on use of the technologies in commercial processes. So, and I have to say that we've certainly gained a lot with collaboration with Corteva in terms of the capacity for improving the transformation efficiencies. In sorghum, there's various vector-free systems and things like that. So it's been very productive.
That's great. So Christine has congratulated you and the team. So that's Christine Beveridge. There's another question that just came in. If this was demonstrated to give the same yield as conventional hybrids, do you think seed companies would take up the methodology?
Yes, absolutely. In fact, what's happening now, I think, is the foundation is considering another phase. I understand that there's what they would like to do, I think, is to consider a proposal whereby the efficiencies of the work in sorghum and the work in cowpea increase the efficiencies of the process to actually make a pre-commercial phenotype. And I think it's going to be a smaller phase that might be 3 or 4 years, and it's going to involve, I understand, potentially Marissa Simon from Corteva and Jean-Philippe Vial-Calzada from Mexico to do some of that molecular manipulation. I understand that they've been asked to submit a proposal. I think the foundation, from the feedback that I have, has been very happy with the tremendous work that's been done on showing heterosis and in cowpea and sorghum to move things forward. They see a value in continuing on later confined trials and things like that, but there's no point really in doing any of that hard work unless you've got something that reaches the efficiency of the sexual. Because no one's going to take it up, are they? Mm.
Another question here is every potential hybrid must undergo knocking of genes to reduce the cost of hybrid seed production. How to make it in a lot of regions specific hybrids in one go?
Oh, I don't know.
Hmm.
I don't understand what knocking.
I'm not sure knocking either. Knocking out, maybe every potential hybrid must undergo knocking of genes to reduce the cost of hybrid seed production. How to make it in a lot of region-specific hybrids in one go. I'm not sure I fully understand the question either.
I think it might be knockout. I don't know. Sorry. Maybe we can get back to you.
Oh, they say knock out the miney. So every potential hybrid must undergo knocking out a miney to reduce the cost of hybrid production. How do you make that happen in a lot of region-specific hybrids in one go? I guess you'd have to do it one at a time.
So we've been discussing this a little bit, and the idea is that if you can make the knockout specific, so for example, if you had it in one parent, this business of equipping one parent and then doing the knockout with another parent, the idea would be to deregulate the sexual and each sexual parent and then use them in crossing so that you could actually not have to deregulate the final. You'd actually be able to have the progeny. I think the logistics of this hasn't really been worked out at this point, and I'm probably not the best person to answer your question. I think it requires a little bit more thought on how the induction is going to work to actually make it as efficient as possible. I don't know, Brett. I think we've certainly been having some discussions with David and others about how to do this, but until we get the efficiency up, that'll drive more on how to actually hone it down to make it breeding friendly, so you can introduce the technology into with other parents.
Yeah, that's right. And I think the technology, just in the biotechnology space in general, it's advancing so rapidly that things we do today will be faster or better in, tomorrow. So these things will become cheaper and quicker with
I
time.
think I think they'll also um, given that for each crop you have its own problems because the genes are that you have to target are a little bit different, which is I think the issue. Um, the methodologies to actually make it helpful might require a bit of thinking across the different people that are working on the crops, right?
Yeah.
And so I'm kind of hoping that there will be more collaboration to get over some of these hurdles, because we all have the same problem. They'll have it in rice, they'll have it, in other places. I mean, it's so I think it's probably something we can collaborate more effectively on in the future, I hope.
I think there's time for just one last question. So the last one is just great work, Anna. Would a Gates Foundation be interested in millet self-reproducing seed for small farmers using the same approach as described in your talk?
I think so, because I think there's been Peggy's been involved in a lot of work with millets, and I, I'm I think these two crops are kind of the first ones off the rank, and then I think it might be possible to then move this into millets. Some of this is also the establishment of these capabilities within different countries in sub-Saharan Africa. So there would be no reason, for an uptake of that in collaboration with others that want to use it. So sure.
That's great. Okay, well, I'm sorry that we can't get to the remaining questions, but because of time. But I wanted to thank again everyone for joining the seminar. Anna, are you able to switch to the next slide?
Oh,
I,
sorry. Yeah, thanks. I just want to thank everybody for attending, um, and certainly we'll try and answer some of the other questions directly that have come through. So thank you very much. And I'll switch to the next slide, she
so yeah, I
says.
wanted to thanks Anna once again for a really stimulating seminar. And I wanted to say the next seminar in the series is on the 14th of July and features Emeritus Professor Robert Henry and is titled Research for Food, Energy, and Conservation of Biodiversity. And then if there's one more slide after this, Anna. Yeah, so if you haven't already, please sign up to the seminar invitation list, which you can scan with the QR code, or you can look it up at quafi.uq.edu.au/science_seminar. And with that, yeah, thank you again, everyone. Thank you to Anna for your presentation, and thanks everyone for joining us.
Thank you, Brett. See ya.